Okay, hello everyone. Good morning. Last Monday I have not meet you because my program, my virtual meeting, those who are newly hired instructors. So we had an oath taking kaya hindi ko na tinuloy yung online class natin. So that is why I not also record, so I will just continue with the lesson what we for the last week. Um, Thank you for joining the class. So I will now share my screen, class. Okay. So we will now we'll now proceed to the lesson today, which is the chronological events of fermentation technology. Okay, before anything else, who among you here likes to eat kimchi? Who among you here likes Korean food like this, kimchi? Do you like eating kimchi, class?
Yes, ma'am.
Wow, very good. So because we are on that Korean or hindi ko alam if you are influenced with the Korean things, but yeah, it's with the beneficial effects this kimchi have. Of course, we love to eat that, no, and we like spicy food, of course. So this is one of the fermented foods that has been developed and also are already famous nowadays. So how about this one? These are fermented foods. Of course, wines, of course, who I think everyone likes to drink wine also because this is fermented wine and it has lots of health benefits that we can really take in, and it's really good for the heart. So when we have occasions, so andito yung may wine talaga na kasama, and um, medyo may kamahalan na nga, of course. Ganun. So, another fermented food. Do you know what's this, class? Do you know what's this food? Is this fermented food?
No, ma'am.
No? So this is a Japanese fermented natto. It just is a soybean. So ang microorganism ano nila ferment is a Bacillus subtilis. So it is a fermented food in Japan. So I've not yet tasted this one, but um, I want to then to taste this kind of food, but wala, hindi pa ako nakatikim nito even when I was living in Luzon, so wala akong naano pa doon sa mga Japanese restaurant. Ganon. So another is that this is a Japanese fermented natto or a bean. Natto is a bean, and ito naman is a Indonesian food which is called tempe. It is also a fermented food, and you know this white covering, this is a mycelium. So ito yung sa molds to, I forgot the name of the microorganism that that has been used for this fermentation, but it's still also a beans. So I think it's a soybeans ito ang ginamit nila. So they have the white mycelium covered the soybeans. So, and I tasted this, and it's really medyo, it has an awful taste, but then since it is fermented and it has lots also of beneficial effects. So ito yung these are some of the products of the fermented, of the fermented foods in different countries, so a lot of people likes to eat these things because of beneficial effects. So these are the products of fermentation. So for today's lesson, of course, we will have a throwback, or we'll go back to the past and see some chronological events of the fermentation technology. And we have this four waves that we can say it, we can term it. There are four waves that happened in the history of fermentation technology. So for the first wave, it is in the pre-1900s, okay? So it was in this time that the first large breweries were created from the early 1700s, when the Egyptians used the first wooden vats of 1,500 barrel capacity for wine fermentation. So in the ancient civilization class, um, fermentation is already taking place because in the from the ancient people, of course, this fermentation has a religious significance, and in Judaism and Christianity. So, but they don't know what the mechanism of why there is fermentation, why there will be fermentation of wine, why they can produce wine. So they have no knowledge, and I think there are no curious people that investigated for these things. And it was also noted that Babylonians also had a history of using of fermenting wine. So these are the people, yung mga Egyptians, mga Babylonians. So they have, I think they are neighboring countries. So yun yung ang kanilang it was really spontaneous. So it was just an accident that they found out that there are their foods are fermenting as they store it, ganun. So yun yung process nila. So because of that, so they found out that they have to store wine, and it's really beneficial into their health. So they have to upscale it, and they have their first large breweries that were created. So by the Egyptians. So yun, nagkaroon sila ng large quantity of production of wine, or the alcohol fermented drink, ganon. So the process of control was also introduced as evidence, showed that they used thermometers and 1757, and the heat exchangers developed in 1801. So they have the kind of idea that they have to control the conditions, such as thermometer, such as the temperature, and also the heat that should be as long as the production of the wine was continued. So ganun yung ano nila, but it was still they have still no idea of what was really happening inside the fermentation media. So what is really happening, what causes the fermentation to take place. Ganun. So it was just a part of their daily activities, that they have to do this and do that. So ganun. That was the first wave. So they have no idea of the mechanism of the fermentation. Okay, so now in the middle of 1800s, of course, yes, I think nakalimot ko class kung kita na mo sa akong slides, no? No, yeah. Kita na mo sa akong slides, class, no? Nakita na sa inyong screen?
Yes, ma'am.
Okay. So in the middle of 1800s, of course, this is Louis Pasteur. So Louis Pasteur was known to be the father of modern microbiology, and he convinced the scientific world the important use of microorganisms in the process, or in the fermentation. So he was really the one who pioneered, or demonstration of the role of yeast in the fermentation. So that's why Pasteur was really had a notable findings, or was really known as the father of modern microbiology, because he demonstrated it clearly. And additionally, also Cagnard, Latour, Swan, and Cosing had also their first investigation about the role of yeast in the alcoholic fermentation. They also have the investigation also, but it was really Pasteur who really had a detailed demonstration, a clear, crystal clear experimentation of the role of microorganism in the fermentation. And also in the 1800s, so they really another scientist also, this person, si Hansen, Emil Hansen, by the late 1800s, he pioneered his work on pure culture isolation. So that was now the beginning of the pure culture technique, that your microorganisms should be purified or monoaseptic, so that when you mix it with your fermentation media, so there's no competing microorganism that will consume your culture media, and also you have that high increase of production of the fermentation. So that is why the Hansen pioneered his work on this pure culture technique course. Yes. So that's it for the first wave. So it was really Pasteur, Hansen. So and the second wave na, it was really the beginning. So they have already, I think the microbial world, the scientists have already, or the people have already convinced, were already convinced to the pure culture inoculation and pasteurization. So pasteurization was also introduced by Louis Pasteur, of course, from his surname Pasteur. So it has to be that the pasteurization has to the what they call that, in order to have a pure culture inoculation, your media has to be pasteurized also. Of course, at pasteurization is the heating of your some would say culture media, or of course milk, like for example milk. So it has to be pasteurized, or it has to be heated at 70 degrees Celsius to 75 degrees Celsius for 30 minutes, so that there will be killing of the microorganisms. And before, for example, for the milk, before you have to use it for the yogurt production. So that's the concept of pasteurization. So and also in the second wave also, there were already products that were produced, like the yeast biomass, the glycerol, citric acid, the lactic acid, the acetone, and butanol. So may mga marami na ang na-produce ng mga fermented products, and these are purified also by microorganisms. These are purified that are produced from the microorganisms because of this pure culture inoculation. So it really paved the way of high increase of production because of the pure culture inoculation technique. So of course, not just there, but also they also purify the glycerol, the citric acid, the lactic acid, the acetone, the butanol, so on and so forth. So marami na ang nangyayari na mga improvement of the technology that in the second wave. So excuse me muna class, because my laptop is already in a low battery, so actually charge. So, okay, let's continue. So, it was also the time that Wisman established aseptic fermentation during the First World War to produce acetone butanol. So, I think that the acetone butanol, because of the high demand of the products, for example, acetone, butanol, siguro it is used for weapons during the First World War. So they have to do improvement, and they also established the aseptic fermentation, in which they had to sterilize muna the culture media before inoculating those microorganisms used for the production of acetone and butanol. So because of this demand, because meron ng war that was happening that time, of course they had to think improvements or ways in order that there will be high increase of production of this kind of products. Okay, so now in the third wave, so it was really a breakthrough, no, for Alexander Fleming, because it was that time that the penicillin was discovered, and not just discovered, but it was really massively produced in submerged culture under aseptic condition because of the antibacterial medicine during wartime. So there was a high demand of antibacterial medicine at that time because it was already in the World War II, and many were wounded, many soldiers are suffering with cuts and wounds and mga shotgun wounds, di ba? So there are many casualties during that time. So they have to find ways of how to cure these wounds. And of course, kung walay antibacterial or walay antibiotics, of course it will get worse because of bacteria that could infect the wounds, di ba? So they have to be taken care of, and they have to be applied with just antibiotics. So it was the time that Alexander Fleming discovered, and also it was really massively produced. Actually, it was the role of the USA, of the United States, of the America, was really the one who funded the large production of this penicillin. So thanks to Alexander Fleming, because he discovered this, and it was just an accident that he discovered that there is a product or a antibiotic that is produced from the Penicillium tutatum, in which it kills the Staphylococcus aureus that time, or the Staphylococcus sp. So with this discovery, of course, and there was a massive production of the antibiotics. So there was really a breakthrough also that the soldiers can be cured already. Those wounded mga civilians and other those patients or who are suffering also with such bacterial infections. So there was already a cure, or also a intervention in order that they will be healed quickly, of course. And then it was also that time that pilot plant facilities were also developed, and also for the massive production of the penicillin. So it was the most significant change in the fermentation technology that results in large production of other important commercial products. So it was the beginning of the antibiotics era, that penicillin was the first to be discovered, and other antibiotics from different microorganisms were also discovered during that time. So until now then, so marami na ding mga antibiotics that are really available now in the pharmacy. So pero hindi natin yan mabibili over the counters. It should be prescribed by the medical doctor. So that's it for the third wave of the fermentation technology. And also for the fourth wave, it was also the use of single cell protein using hydrocarbon and other feedstocks that was introduced to improve the production of products. So like single cell proteins such as spirulina, used for the supplementation. So in the development also of the fermenter, There were also pressure cycle and pressure jet vessels that were developed in order to overcome gas and heat exchange problems and was computer operated. So it was also the beginning of the computer operation for the fermenter, so the bioreactor. So later we will discuss about the different parts of the bioreactors. We will also discuss the different function, uses and function of the bioreactor in order to produce those products that are really used. So these are the bioreactors are used for the upscale of the production of that particular fermented product. Ganun. So additionally, to minimize the human error of the large production in the fermentation, so the computer system was already utilized and established, of course, to perform fermenters aseptic operation. Because when there is, if we use still the manpower operation, so there are many errors that can be committed because of different because of course the human resource are really subjective sometimes, and maybe there are maybe also when they really do the experimentation and of course establishment of the condition, they might forget it. So ganun. So we have to improve the facility by using computer generated systems so that to have a memory, yung stored memory na yung mga conditions in the computer. So, yung ginagawa na lang ng mga scientists or mga operators, they just have to input the condition and also just input the condition, of course, control, and also monitor the process, ganun. So, about the temperature, the pH, ganun. So, less na lang yung ginagawa ng mga tao. So, ganun. This is really helpful din sa fermentation industry because it won't take them a lot of it won't give them I mean there is reduction of cost when there is reduction of cost, or they won't hindi sila walang masasayang na mga production media or ganung mga fermentation culture when there is error that will happen because of these computer-generated systems na. So, yun ang fourth wave. So, as you can see that during the first, so there was no idea of the fermentation that take place, and it was just a part of the civilization in the lives of the Egyptians, Babylonians, and we don't know yet for other mga ancient people from the other countries. So maybe they were also doing that. So now when this French microbiologist in the second wave, of course, Louis Pasteur has introduced that there is a role of microorganisms, and also he demonstrated it in a experimentation, and he also introduced pasteurization, ganon. So, and then the third wave it was really the beginning of the pure isolation technique, ganon. And also, it was the second wave, no? Tama, that it was the beginning of the pure isolation technique, and also in the third wave, it was also the beginning, the booming of the antibiotic industry that began by Alexander Fleming. And here also in the fourth wave, it was already the computer digitalization. So yun yung mga nangyayari na ngayon. And until now also, if you have, I've also visited such mga industrial companies like Yakult, so saan pa ba? Like San Miguel, we visited there. in Batangas. So marami na talaga ang improvement, and then they have a large pilot plant facility. They are really doing well because of course they are producing different kinds of alcohols, different kinds of drinks, and also they also supplying alcohol talaga, as in alcohol for the disinfection. So the role of the industrial company last year, 2020, because of this onset of the pandemic, they really had a double or shall we say triple na production of alcohol. So they really played a significant role during the onset of the pandemic because they were the ones who are really producing the alcohol for us so that we can really use it on a daily basis and of course prevent from infecting the virus. So yun ang that's the summary or the notable history of the fermentation technology. And I guess that there are a lot of things pa din that happened in between because this actually mahaba siguro talaga if you just want to take details of this fermentation technology, and there are a lot of interesting events that happen. So later in the learning task, I will give you some tasks to research interesting events about the history of fermentation technology. Okay? So sa fifth wave naman, so there are fifth wave, hindi pala siya 4 waves, but 5 waves pala class. So there is now the production of heterologous proteins and microbial and animal cells that was started. And there are also monoclonal antibodies that were produced. So ano ba yung monoclonal antibodies class? These are antibodies that was extracted from the mouse. Of course, these could be human antibodies that was produced from the mouse. And these monoclonal antibodies will be subjected to the human trials. Of course, it's used for for example, the infection of such diseases or the viruses. So these are the things that was already produced during the fifth wave. So it was in the 1979 up to date. And there are also different kinds of organisms that were initiated and developed. So marami na din talaga ang na-develop ng mga organisms and na-use ng mga organisms for the different kinds of production of citric acid, acetone, different kinds of proteins, yeah, and also the breakthrough of the recombinant DNA technology. It was really the advent of the new genetic engineering techniques. So in order for us to discover natural products and also to design drugs that was really first introduced by the established pharmaceutical industry. So during this fifth wave so they really had an investigation in how to clone a gene in order, like for example, for the insulin na pwedeng mag-produce ng higher amounts of insulin through a microorganism like E. coli, like Saccharomyces cerevisiae. So ganun yung mga ginawa nila. So they have to clone the gene, take it out. They have to cut the gene from the mammals, of course, and then also they have to transfer it. They pasted it in the plasmid of an E. coli and then introduced the plasmid of the E. coli and then transform. Okay, and they have to see the expression if the E. coli produces insulin, of course. So this was really possible at that time, no? So later on, when you reach your InBt124 for the second sem, you will really appreciate the different history and really the different techniques. It was really mind-boggling, of course, class, but it was really na-appreciate ko talaga ng maganda when I really also when I also proceed in my MS studies. Of course, it was really hard, but you really appreciate all the more the techniques of the molecular biology and biotechnology here. So as we all know that these things or genetic engineering are really important and played a significant role in this pandemic, and also it will also play a role in the near future, of course. So that is why we're really in demand. So your life class are really important because you have a future to do and to really to take place in the country or in other country or in this kind of in your generations to come. So that's it for the fifth wave. So yun ang five waves ng history of fermentation technology. So there are also component parts of the fermentation. Of course, there are many different components lang, hindi lang yung microorganisms, kundi meron din conditions that has to be controlled, that has to be established in order to have to achieve higher production of these products or fermented products. Okay, number one is the formulation of the media. Of course, what kind of media or the nutrients of the microorganisms, of course. So the food of the microorganisms. So in other words, okay, the food of the microorganisms. So you have to establish what kind of food you have to nourish your microorganism so that it will grow and so that it will initiate the production of your particular product. Ganon. So there is there's an inducer or initiation that there will be high amounts of production of your particular product. So you have to have a carbon source, nitrogen source, oxygen source, and many more. So these are the components of your mga carbon source, of course. Yeah, the formulation of your media. You can use glucose, you can use yeast extract, ganon. So later on, we'll tackle about the formulation of the media and how to optimize all these media components in order to have a to achieve higher amounts of production in the media. Okay, it is used for the process of the microorganisms and also for the preparation of the inoculum. So before you have to mix or inoculate your microorganism in your fermenting media, in your fermentation, you have to establish the microorganisms to be active in state. It should be in a dividing stage or in the log phase of your of the growth of the microorganisms. You don't have to, for example, plasma, that your microorganisms has been stored in the refrigerator. So it should not be, it is not recommended that you just have to take out your microorganisms in the ref and then you just have to inoculate that in your fermenting media, because your microorganisms some of your microorganisms are already dead, and also they are not in the stage of the actively dividing stage of its growth. So and then also the media there in the for example in the test tube or in the fermentation earlier flask. So we can say that there is now a depletion of your nutrients. So there is a high possibility that your microorganisms will not perform or not produce high amounts of your production of your particular product. Of course, that's one of the consideration, So that is why you have to prepare your media, of course. For example, if it's small, you have to prepare PDA, potato, dextrose, agar. There are pre-mixed media that are available in the different supplier companies. There are pre-mixed naman, no? So that you can really establish the condition of your microorganism before inoculating that in your fermenting media. Of course, that's one of the consideration that you have to do. Another thing, the component and the parts of the fermentation is your sterilization. So sterilization really is the killing of the unwanted microorganisms that will participate also, that can compete in the consumption of your nutrients of the particular fermentation medium. No, so you have to sterilize your medium. You have to sterilize also your flask, of course, so that there will be no, of course, the sterilization covers also the killing of the pores of the molds, ganon. So and also the sterilization of the medium, the fermenters, and the ancillary equipment. Those accessories that should be used, no, for the fermentation. Of course, we don't want to that your media has an unwanted microorganisms that can compete for the production of such particular products. Also, it is used. This sterilization can be done using autoclaves for the assurance of pure culture fermentation. So later on, we'll also discuss about the topics of sterilization technique. So another one is the culture of an active pure culture that is that has a sufficient quantity to inoculate the production vessel known as the seed culture. So ganon. So this is the seed culture, which is you will establish the condition of your microorganisms. So iba din yung the seed culture will serve as your inoculum. Okay, so that you will place this in your media that has a higher volume of culture media, and it will now be used as uh, the vessel for the fermentation talaga in order to produce high amounts of this particular product that you want to produce. Okay, so sorry. Okay, another component is the optimized growth condition of the microorganism. So you have to know the optimized temperature, the optimum pH, the optimum incubation period, the optimum agitation or the RPM if your microorganisms is aerobic. If you will do aerobic fermentation, of course, you have to agitate it. You have to investigate it through a optimization experiment. You can also do variable screening experiments. So later on, I will also introduce to you how to do that in an experimental setup, because I think I have to tackle this topic for you, because it is really significant that we will have to know how to get the optimized conditions of your microorganisms. So ayun, yun yung mga conditions that, of course, the microorganisms are really sensitive, and some microorganisms are really sensitive in temperature, in light, and also in pH and agitation. So you have to consider all those components so that your microorganisms will have a favorable production of the particular product that you wanted. And also, you have to consider also the extraction of the product. So it is now in a downstream processing. So katong akong ipang-discuss recently was really the upstream processing. So for the extraction of the product of course, you have to extract your product. So you have to know if your product is an extracellular product or an intracellular product. So these are two different things, because with this identity of your product, it will also determine what kind of technique you have to do for this downstream process for the extraction of the product. No, if it is extracellular, then you are not worried, because you just have to separate the cells and also the media. Of course, the culture media. Of course, the product that you wanted are in the culture media because it's extracellular, because the microorganisms secrete out of the cell. While if it is intracellular, so the microorganisms will produce inside the cytoplasm, in the cytoplasm of the cell. So when you extract your particular product, you have to lyse the cell, or you have to kill the microorganism so that your product or your particular protein will be excreted out of the medium. So yon, dun na ang mga, and then you'll have many different degrees, proteins and DNAs that are already mixed in your culture media. So hindi lang, you don't expect na sa imong culture media kay naranadiha dayon ang imong your product is alone, chemical also is present in the culture media. So you have to purify it, you have to do mechanical techniques about it. So later on we'll discuss it, no? And last thing is the disposal of the waste produced by the process. There are many waste also that that are not part of your purification. So when you go through purification techniques, there are waste that you have to discard. So you have to know how to discard it, and you also have a knowledge about the different products that are also present other than the wanted products that you wanted to purify. Okay, so is it clear? So yun ang different components, parts of the fermentation. So this is just the general schematic presentation of a typical fermentation process. So class, muna siya class. So here in the inoculum development, you have a stock culture usually. Others are these stock culture are stored in the refrigerator, but it is recommended that you have to you have to store it in a biofreezer so that there is low chance of the mutation of your microorganisms. So you have to store it in a biofreezer. Of course, you have to do glycerol stock for this to maintain the viability of your microorganism. No, so the stock culture, and then when you take it into the ref, you have to culture this, and then you have to in a shake flask, of course, to revive. This is so-called revive the microorganism. You have to revive your microorganism in a shake flask. You have to inoculate. So depending upon the optimized conditions, if it is aerobic microorganism, you have to agitate it in order to revive and to have an active stage of growth of your microorganism. So after that, the shake flask, so after it, that you have now achieved the cell concentration of your shake flask, you now have to place it in the seed fermenter course, seed fermenter. So as you observe class, this is gradual na inoculation of in the mixing of the fermentation media, no? Seed fermenter, ganon. So, of course, before that, you have to sterilize your medium, and also there are medium formulation then that you have to do, and then also there are raw materials that you have to acquire. Sorry. Yeah, so you have to do that in the seed fermenter. Actually, the seed fermenter is just a low volume, or the low volume, or the small volume of your production fermenter. Of course, you have to gradually place the microorganisms in small volume before placing it in a large quantity of your fermenter. So there's really a difference class. So when you really have to, and also class na ang cell concentration din sa microorganisms will also vary. So that is why you have to do it gradually. Of course, yeah, that's it. Before you have to place it in the production fermenter. So after fermentation, of course, you have to separate it. So it is now in the downstream processing. So you have to separate the cultural fluid, and then you have the cell separation. Like if you like to have the biomass, also you can also that, and then if the product is an extracellular product, so you have to obtain the cell-free supernatant. But if your product is an intracellular one, you have to also obtain the biomass, of course, and you have to lyse the cell before the extraction of your product. So if it is extracellular, so there's not a product extraction, and then product purification, and then the waste treatment, the effluent treatment, and then the product packaging. So before it is now, and then after that product packaging, of course, you have to test it, quality control, ganun, before distribution to the pharmacies or many stores that can be purchased by the consumers. So yun ang summary or the schematic presentation class of the component parts of the fermentation. So later on, we will also discuss this one by one in order to have a wider grasp and understanding of the development or the important parts of the fermentation techniques. Okay, so I guess, yeah, may learning task pa, but then before I will discuss with your learning task, meron ba kayong question class? Am I too fast?
Okay ra, ma'am.
Okay, Ran. Did you understand class?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so I will now present to you the learning task. So this is now the learn task or activities. Okay, so number one, you have to research an interesting history or event about the development of the fermentation industry that was not discussed from the lesson today. So I can think there are many things. So just one interesting event or history. Class no, inyong that you have to discuss or you have to write it. So, and then second question is to differentiate the word processing and fermentation in your own words. Okay, so that's it for the learning task class. Okay, so in order to submit this learning task or activities class, you have to write it in a sheet of paper with a sheet of paper, and then you have to take a photo of it, and then you have to convert it in a PDF form, and then submit it in the via CEE. Of course, I will provide an icon there that you have to submit your learning task there. Okay, so again, the learning task should be answered in a paper. You have to write it talaga with ball pen, paper. So in other words, it is not encoded. Okay, it is not encoded. Kailangan siya na isulat talaga. And then you have to take a photo of it and then convert it into PDF form and then submit it in the VSUE. So you have 3 weeks to accomplish this task. It's just so easy class. So it's just you have to research it naman talaga, of course, yeah. So that's the learning task for this lesson. So that's it, okay? So I think I will just give you the post test siguro na lang next week before we begin our next lesson class. Okay? So did you understand?
Yes, ma'am.
Yes, ma'am.
So, is there any question? It's already 11 a.m. Before anything else, class, is it okay with you na may klase 11 a.m. to 12 a.m. Monday or Wednesday? Na may klase?
Wala, ma'am.
Wala mo tanan. So, is it okay with you that we will just meet 10 to 12 a.m. in one, kanang 12 noon on Wednesday para dili na lang ta mag-meet sa Monday 10 to 11? Okay ra ninyo
10 to
class?
12 na to ang Wednesday, ma'am.
Ha?
10 to 12 ang Wednesday.
Oo.
May 2 hours.
Para once a week na
Okay.
lang ta mag-meet.
Okay ra ko, ma'am.
How about the others?
Okay ra sad, ma'am.
Okay ra sa
Okay ra
inyo?
siya, ma'am.
Oo, oh, okay, sige. It's already official that we'll just meet once a week pero sa Wednesday kay 10 to 12 na ta. So diretso-diretso ng atong klase para once a week na lang kita. Para ning Monday dili na lang ta mag-meet. Okay, kay para 2 hours na lang diretso. Okay? So and also class I have posted in the student I posted in the VSUEE that you have to accomplish the student honor pledge. You have to furnish it, and then you have to sign it with your signature in order to you have to read it naman, and then as a pledge that you will be doing with honesty and integrity in this course. So and also I would like to ask your grade in BTEC 22. Just submit it in the na lang sa VSUEE. I have also provided that doon sa just visit the VSUEE class. Okay? So I give you 1 week naman to accomplish it. So just ano lang, if you have now the time and it's favorable with you. Okay? So do you have any questions before I will leave or wala na?
Ma'am, nag-ask na ko na classmate, ma'am, if asa daw, atong wala maka-attend pa, ma'am, asa daw nila pwede ma-access ang lecture karon.
Yes. Actually, I just uploaded it last night, so I have not yet posted it in the VSUEE. So, mamaya I will just post the link of the first lecture. So, just antabayanin nyo na lang. I will just inform you. I will just chat you in the group chat if na-post na. Okay?
Okay, ma'am, so sa VSUE ra na mo mapaan.
Oo, sa BSCE. Okay,
Thank you,
so
ma'am.
welcome, Ariel. So, is there anything else? Wala na?
None, ma'am.
Ano, ma'am?
Nan Nana Rashid? Okay, if there is none, so thank you so much for joining the class. So, see you next Wednesday, 10 a.m. to 12 noon. Okay? So, I think bukas may Thursday na class, which is our laboratory in InBt121. So, it's 8 to 10, okay? So, it's 8, bukas we will have a lab class, 8 to 10. Okay, so thanks class for joining. Have a blessed week. Bye-bye. God bless.
Thank you, ma'am.
Bye-bye.
Thank you,
Thank you,
ma'am.
ma'am. God
Thank
bless.
you
Thank
so
you, ma'am.
much.
Thank you, ma'am.